Although Louis Pasteur and Edward Jenner developed the first vaccine to protect against viral infections, they didn't know that viruses existed. The first evidence of the existence of the virus came from experiment with filter that had pores small enough to retain bacteria. In 1892, Dmitry Ivanovsky used one of these filters to show that sap from diseased tobacco plant remained infectious to healthy tobacco plant despite having been filtered. Matrina's visionary called the filtered infectious substance a virus, and this discovery is considered to be the beginning of virology. The subsequent discovery and partial characterization of bacteriophage by Tewart and Harrell, and by the early 20th century, many viruses had been discovered. Thomas Milton River defined virus as obligate parasite. Viruses were demonstrated to be particles rather than fluid. Stanley and Raska invention of the molecular microscope in 1931 allowed their complex structure to be visualized. Pasteur was unable to find causative agent for rabies and speculated about a pathogen too small to be detected using microscope. In 1884, French microbiologist Charles Chamberlain invented a filter known today as Chamberlain filter that had pores smaller than bacteria. He could pass a solution containing bacteria through the filter and completely remove them from the solution. Meyer, who directed the agricultural experimental station in Wageningen, was the first who showed that tobacco mosaic disease was infectious. He thought that it was caused by either a toxin or a very small bacterium. Later in 1892, Russian biologist Dmitry Ivanovsky used a Chamberlain filter to study what is now known as tobacco mosaic virus. His experiment showed that crushed leaf extract from infected tobacco plant remained infectious after filtration. 1898, Dutch microbiologist Matrina Springiek, microbiology teacher at the agriculture school, named repeated experiment by Adolf Meyer and became convinced that filtrate contained a new form of infectious agent. He observed that the agent multiplied only in cells that were divided, and he called it contagium vivum fluidum, soluble living germ. The visionary maintained that viruses were liquid in nature, a theory later desecrated by the American biochemist and biologist Stanley, who proved that they were in fact particles. In same year, Loeffler and Fuchs passed the first animal virus through a veterinary virology. Welcome. Let us read this photo. Infectious disease is one of the few genus and nature left in the world. The dangerous are all dead, and the land grows rusty in the chimney corner. About the only sporting proposition that remains unimpaired by the relentless domestication of one free living human species in the world remains those species little fluid creatures which lurk in the dark corners and stalk us in the woods of rats, mice, and all kinds of domestic animals which fly in the field with the insects and wildly us in our food and wailing us in our food and drink and even in our love. This photo is taken from the book Rats, Lice and History, written in 1935 by the great microbiologist and themselves as he reflected in his life in infectious disease science. Let us do the definition of the virus simply. What is virus? Virus may be defined as a cellular organism whose genome consists of nucleic acid and which obligately replicate inside host cells using host metabolic machinery to different extent to form a ball of components which assemble into particles called virion. Viruses can't be grown on sterile media but require the presence of specific host cells. A virus differs from a cell in three fundamental ways. A virus usually has only a single type of nucleic acid serving as its genetic material. This can be single or double-stranded DNA or RNA. Viruses contain no enzyme of energy metabolism, thus can't make ATP. Viruses don't encode sufficient enzymatic machinery to synthesize their component macronucleus, specifically no protein synthesis machinery. Fundamentally, the virus is a package of genetic information protected by a protein shell for delivery into a host cell to be expressed and replicated. Viruses are distinguished from other obligate parasites, some of which are even simpler than virus, mycoplasmas or bacterium that grows on complex media and attached to eukaryotic cells. Chlamydia, obligate intracellular bacterial parasite which depend on eukaryotic cells for energy. Protozoa, obligate intracellular parasite that replicate within eukaryotic cell. Varioid, infectious agent of plant that exists as native nucleic acid, therefore single-stranded RNA. Hepatitis delta virus, HDV, viroid-like agent whose replication is dependent on HB hepatitis B virus. And this is Stanley Berger, the winner of the Nobel Prize in 1997 for his discovery of barium. Barium, pernicious infectious agent, hypothesized identity of the unconventional slow virus such as cold, scurvy, and gout disease agent. No nucleic acid is known to be required for barium function. They are caused by needing to consist slowly of protein and perhaps diseases caused by barium or barium diseases. Creutzfeldt-Jakob variant, Creutzfeldt-Jakob, Strauss-Love, and Schinker syndrome, fetal familial insomnia, and corona. And this is the histopathology of some of these diseases: scurvy, DSA, bovine spongiform encephalopathy, variant Creutzfeldt-Jakob. The recognition of virus. How long viruses have been within our 1500 BC, leg deformities indicative of poliomyelitis and bookmarks indicative of a small flavero. Siltah ruled Egypt from 1200 to 1193 BC, when he died suddenly at the age of about 20. His mummified body lay undisturbed within his tomb in Valley of Kings until 1905 when the tomb is excavated. The mummy shows his leg was withered, his foot was strangely extended like a horse hoof. The pharaoh Ramses V, who died in 1196 BC, is believed to have succumbed to smallpox compared to the pustular lesion on his face and his lip. During the age, all infectious agents were considered to be virus until Koch developed pure culture technique which allowed the separation and growth. Bacteria were verified and established as disease-causing agents, and then became possible to distinguish them from filterable agents, those able to pass through a special filter designed to prevent the passage of bacteria. And this filterable disease, the coronavirus in 1898, yellow fever, flavivirus in 1900, Rao sarcoma virus, and oncogenic retrovirus in viral disease have played a major role in human history over whatever time scale we choose to explore. Over the past 1,000 years, smallpox and measles were brought to North and South America by early European explorers and conquerors. These diseases, for which the Native American population has no acquired partial immunity, killed large fractions of the populations and were a major factor in the dissemination of these societies. As the century entered its final 20 years, new 100 lethal virus HIV spread rapidly around the world via body fluid transmission. Over the past 100 years, a newly emerged strain of influenza killed 20 million people in 1980 and 1990 in the immediate aftermath of World War. And this is picture displaying. A decade later, polio became one of the most feared infectious children and young adults, proving Franklin Roosevelt, the US president, throughout Depression and World War II. The global HIV epidemic continues. Sporadic cases and outbreaks in human of some non-human viruses such as Ebola and Hanta raise the concern about future epidemics by other viruses in new century. Coronavirus, severe acute respiratory disease, SARS, is a contagious and sometimes fatal respiratory illness. SARS first appeared in China in November 2200. Within a few months, SARS spread worldwide, carried by unsuspecting travelers. SARS showed how quickly infection can spread in highly mobile and interconnected coronavirus disease in 2009. COVID-19 is a contagious disease caused by virus, severe acute respiratory syndrome, coronavirus 2. The first known case was identified in Wuhan, China in December 2019. The disease quickly spread worldwide, resulting in COVID-19 pandemic. The discipline of the virology, the study of virology inherent, involve a merging together of what has traditionally been thought as two separate kinds of science, basic and applied science. We want to figure out how viruses are transmitted, how they replicate, and how the host organism respond. We also want to figure out how to prevent transmission, how to interfere with the virus replication, how to confer immunity on the host. The applied is for and is dependent upon the basic in virology, as it is studied today, is therefore an outgrowth of both the infectious disease and the molecular biology. Infectious disease because of the recognition of the viral pathogens, molecular biology because of the usefulness of the virus as probes of cells, and molecular biology and metabolism, and as a vector with a strong potential of gene therapy. This is the primary, mouse primary living cell transfused with a green fluorescent protein, retrovirus vector. Viruses are the smallest of all the microbes. They are said to be so small that 500 million virus which cause the common cold could fit on the head of a bird. They are unique because they are only alive and able to multiply inside the cell of other living things. The cell they multiply in is called the host cell. Virus structure. A virus is made up of a core of a genetic material, either DNA or RNA, surrounded by a protective coat called capsid, which is made of protein. Sometimes the capsid is surrounded by additional spike coated called envelope. Viruses are capable of latching onto the host cell and getting inside them. Acquired immunodeficient virus or AIDS, which chiefly retrovirus, which named immunodeficient virus HIV, is a typical virus to study the structure of the virus because it has many details. This is a core RNA virus, RNA, and there is enzyme reversing transcriptase, there is the matrix protein, and this is the lipid membrane, and this is the spike GB120, GB41, and there is the capsid. A full structure virus, and it has many details. Classification is the process of naming viruses and placing them into a taxonomic system similar to the classification system used for cellular organisms. Viruses are classified by phenotypic characteristics such as morphology, nucleic acid type, mode of replication, host organisms, and the type of disease they cause. The formal taxonomic classification of the virus is the responsibility of the International Committee on Taxonomy of Virus, although the Baltimore classification system can be used to place virus into one of seven groups based on the manner of messenger RNA synthesis. Specific naming conventions and further classification guidelines are set out by the ICTV. The International Committee on Taxonomy of Virus began to devise and implement rules for the naming and classification of virus early in 1970s, and F union from microbiological societies with the task of developing, refining, and maintaining a universal virus taxonomy. The system shares many features with the classification system of cellular organisms, such as taxon structure. However, some differences exist, such as the universal use of italic for all taxonomic names, unlike in the International Code of Nomenclature for algae, fungi, plants, and the International Code of Zoological Nomenclature. International Code of Zoological Nomenclature is the phylum, subphylum, kingdom, subkingdom, and order and family. Baltimore classification, first defined in 1971, is a classification system that places virus into one of seven groups depending on combination of nucleic acid, DNA, RNA, stranded, single-stranded, or double-stranded sense, and method of replication named after David Baltimore, Nobel Prize-winning biologist. Double-stranded DNA viruses like adenovirus, herpes, and pox. Single-stranded DNA virus, positive sense strand, the DNA parvovirus. Double-stranded RNA virus like example retrovirus. Single-stranded RNA virus, positive strand RNA virus like coronavirus, coronavirus, Tuga virus. Negative sense single-stranded RNA virus, negative stranded and sense RNA, also exoviruses, rhabdoviruses. Single-stranded RNA RT viruses, positive stranded sense RNA with DNA intermediate in life cycle, retrovirus. Double-stranded DNA RT virus, DNA with RNA intermediate in life cycle, hepatinovirus. Virus family, example virion and the capsid symmetry nucleic acids group, adenovirus. Adenovirus like canine hepatitis virus. Some types of common cold, the virion naked, and the capsid icosahedral, nucleic acid double strand, bapuvavirus, parvovirus, herpes virus, pox virus, and
example of the DNA viruses, example of RNA viruses, reovirus and rotavirus, the capsid naque, the capsid symmetric hetero, bicorna viridi, calciviridi, togaviridi, arenaviridi, flaviviridi, orsomixoviridi, bramixoviridi, boniaviridi, rhabdoviridi, and flaviviridi, coronaviridi, astroviridi, bernaviridi. These are examples of RNA virus. Example of reverse transcribing virus. Viruses must first get into the cells before viral replication can occur. Through the generation of abundant copies of the genome and packaging these copies, the virus continues infection new hosts. Replication between viruses is greatly varied and depend on the type of gene involved in them. Most DNA viruses assemble in the nucleus, while most RNA viruses develop slowly in cytoplasm. Tell me more, please. Viruses multiply only in living cells. The host cell must provide the energy and synthetic machinery and the low molecular weight precursor for the synthesis of viral protein and nucleic acids. The virus replication occurs in 7 stages, namely attachment, entry, uncoating, transcription, messenger RNA protection, synthesis of the virus component, virion assembly, and viral replication of the bacteriophage, release, liberation stage. Attachment is the first step of viral replication. Virus attaches to the cell membrane of the host cell. It then injects its DNA or RNA into host to initiate infection. In animal cells, these viruses get into the cell through the process of endocytosis, which works through fusion of the virus and fusion of the viral envelope with the cell membrane of the animal cell. An implant cell, it enters through the process of pinocytosis, which works on pinching of the viruses. Entry. The cell membrane of the host cell invaginates the virus particle, enclosing it in pinocytotic vacuum. This protects the cell from antibodies, like in the case of HIV virus. Uncoating, cell enzyme from lysosome strip off the virus protein coat. This release or render accessible the virus nucleic acid or genome. Transcription, messenger RNA protection. For some RNA virus, the infecting RNA produces messenger RNA, which can translate the genome into protein product. For virus with negative strand RNA or DNA, viruses are produced by transcripting. Then translation. The messenger RNA is used to instruct the host cell to make virus component. The virus takes advantage of the existing cell structure to replicate itself. Synthesis of virus component. The components are manufactured by the virus using the host existing organelles. Viral protein, viral messenger RNA, is translated on cellular ribosome into 2 types of viral protein. Structured protein, which make up the virus particle. Non-structured protein not found in the virus particle, mainly enzyme for virus genome replication. Viral nucleic acid genome replication. New viral genome are synthesized. Template are either the parental genome or newly formed complementary strands. In the case of single-stranded genome, these genomes are made by either a viral polymerase. In some DNA virus, a cellular enzyme, particularly in rapidly dividing cells. Virion assembly. A virion is simply an active or intact virus particle. In this stage, newly synthesized genome, nucleic acid, and protein are assembled to form a new virus particle. This may take place in the cell nucleus, cytoplasm, or plasma membrane for most developed viruses. Release, liberation stage. Release or revelation stage. The virus now being mature are released by either sudden rupture of the cell or gradually extrusion to force out of the envelope virus through the cell membrane. The new virus may invade or attack other cells or remain dormant in the cell. In case of bacterial virus, the release of propionyl variant takes place by lysis of the infected bacterium. However, in case of animal virus, the release usually occurs with cell lysis. Viruses are classed into 7 types of genes, each of which has its own families of virus, which in turn have different replication strategies. Themselves, David Platimore, a Nobel Prize-winning biologist, devised a system called the Platimore classification system to classify different viruses based on their unique replication strategy. There are 7 different replication strategies based on this system: Platimore class 1, 2, 3, 4, 5, 6, 7. The 7 classes of this replication strategy. Class 1, double-stranded DNA. Virus of this type usually must enter the host nucleus before it's able to replicate. Some of these viruses require host cell polymerases to replicate their genome, while others, such as the DNA genome, this linear double-stranded DNA, the family Adenoviridae, linear double-stranded DNA, naked icosahedral virion, structure, capsid, protein 2, protein 3, pentose-base protein, protein 3A, protein 4, protein 6, 8, 9. This is the protein link the DNA to the pentone-based protein 3, protein 7, protein 10, terminal protein, double-stranded inverted terminal repeat sequence. Its structure, the virus structure, 70 to 100 nanometer diameter, protein 2, 3, 3A, 4, 6, 8, 9, protein 7, protein 5. This is the terminal protein, the attachment and the entry. The fiber protein binds the coccivirus adenovirus receptor. Integrin bind the RGD sequence in the pentose-base protein 3. Virus particle escape from endosome. Microtubule bind hexone and transport the viral DNA enter the nucleus, and terminal protein bind the nuclear matrix. Early transcription. The genome is separated into the R strand and the I strand. RNA polymerase 2 transcribe the viral messenger RNA. E1 gene product. The E immediate early bind of the host regular protein. That one inhibits cellular apoptosis. E1 inhibits bind retinoblastoma tumor protein, and this is the messenger RNA gene product. E4 gene product. Messenger RNA contribute to DNA synthesis. Messenger RNA E2 gene product. 3 protein important in DNA synthesis. Gene E3 gene product. Modification of the host immune system. DNA replication occur when the cell move into S shape. The inverted terminal repeat serve as origin of the replication. Terminal protein and the viral DNA protease from E2 region start the replication. Either strand of the genome can be displaced. Transcription, late transcription and translation. Late transcription and translation. Delayed early gene product transactivate late genes. Messenger RNA splicing and different poly A sites product various assembly. The last stage. DNA enter the capsid and get out of the cell. Coronaviridae, positive sense single-stranded RNA enveloped helical. Here is the membrane protein, hemagglutinin, strays, spike protein, flexible helical nucleocapsid protein, positive sense single-stranded RNA. Life cycle of Coronaviridae. Attachment and entry. The spike protein facilitates attachment of the virus to variety of hosts. Virus may enter by membrane fusion or by biocytosis. Primary translation. Neither primary transcription. Discontinuous negative strand transcription. Protein synthesis, assembly and release. Family Flaviviridae. Virus structure. Membrane protein, E protein, capsid protein, E protein, capsid protein, core, positive sense single-stranded RNA, 5 capped. Flaviviridae life cycle. Attachment and entry. Protein sensitive. Genome replication. Assembly and release. Orthomixoviridae family. Orthomixoviridae structure: hemagglutinin membrane protein, hemagglutinin protein subunit HA1 HA2, neuraminidase protein Na, ion channel protein, matrix protein, ribonucleoprotein complex, negative sense single-stranded RNA, nucleocapsid protein, polymerase protein A, polymerase protein A, polymerase protein BB1, polymerase protein BB2. Family Orthomixoviridae and entry. Transcription. Protein synthesis. Genome replication. Assembly and release. Paramixoviridae structure: membrane protein, fusion protein, hemagglutinin neuraminidase protein, matrix protein, nucleocapsid, negative sense single-stranded RNA, nucleocapsid protein, large protein, phosphoprotein. Paramixoviridae life cycle: attachment and entry. Messenger RNA transcription. protein synthesis, genome replication, assembly and release. Viral pathogenesis. Viral pathogenesis is the study of the process and mechanisms by which viruses cause diseases in their target hosts, often at the cellular or molecular level. It's a specialized field of study in virology. Pathogenesis is a qualitative description of the process by which an initial infection causes disease. Viral disease is the sum of the effect of viral replication on the host and the host subsequent immune response against the virus. Viruses are able to initiate infection, disperse throughout the body, and replicate due to specific virulence factors. There are several factors that affect pathogenesis. Some of these factors include virulence, characteristic of the virus that is infecting. In order to cause disease, the virus must also overcome several inhibitory effects present in the host. Some of inhibitory effects include distance, physical barrier, and host defense. The inhibitory effect may differ among individuals due to the inhibitory effect being genetically controlled. Viral pathogenesis is affected by various factors: transmission, entry, and spread within the host, tropism, virus virulence, and disease mechanisms, host factor, and host defenses. Mechanisms of infection. Virus need to establish infection in host cell in order to multiply. The virus has hijack host factor and evade the host immune response for efficient replication. For infection to occur, viral replication frequently requires complex interaction between the virus and the host factor that may result in deleterious effect in the host, which confer the virus its pathogenicity. And its pathogenicity factor affecting pathogenesis: virus tropism, virus factors, host factors. Molecular basis of virus tropism. Virus tropism refer to the virus preferential site of replication in the secret cell type within an organ. Virus tropism refer to the virus preferential sites of replication in the secret cell type within an organ. In most cases, tropism is determined by the ability of the viral surface protein to fuse and bind to surface receptor of specific target cells to establish infection. Thus, the binding specificity of the viral surface protein dictates tropism, as well as the destruction of the particular cell population, and is therefore a major determinant of the virus pathogenesis. Viral tropism can also be governed by other intracellular factors such as tissue-specific transcription factors. Virus factor. Viral genetics encoding viral factor will determine the degree of viral pathogenesis. Different virus strains possessing different virus factors can lead to different degrees of virulence. Host factors. Viral pathogenesis is also largely depend on the host factor. Several viral infections have displayed a variety of effects ranging from asymptomatic to symptomatic or even critical infection. Age and immunocompetence play an important role in dictating whether the viral infection can be modulated by the host. Immunocompromised individuals due to existing illness may have a defective immune system, which makes them more vulnerable to damage by the virus. Disease mechanism. A viral infection doesn't always cause disease. A viral infection simply involves viral replication in the host, but disease is the damage caused by viral multiplication. An individual who has a viral infection but doesn't display disease symptoms is known as a carrier. Damage caused by the virus. Once inside the host cells, virus can destroy cells through a variety of mechanisms. Viruses often include direct cytopathic effect to disrupt cellular functions. This could be through releasing enzymes to degrade host metabolic precursors or releasing protein that inhibit the synthesis of important host factor protein. Viral protein of herpes simplex virus can degrade host DNA, inhibit host cell DNA replication, and messenger RNA transcription. Poliovirus can inactivate protein involved in host messenger RNA translation without affecting polio messenger RNA translation. Persistent infection happen when cells continue to survive despite a viral infection and can be further classified into latent. Lytic virus are shed at the high titer for rapid infection to a secondary tissue host, whereas persistent virus undergo shedding at lower titer for a longer duration of transmission, months to year. Lytic viruses are capable of destroying host cells by curing and or interfering with the specialized functions of the host cells. Damage caused by host immune system. Sometimes instead of cell death or cellular dysfunction caused by the virus, the host immune response can mediate disease and excessive inflammation. The stimulation of the innate and adaptive immune system in response to viral infection destroy infected cells, which may lead to severe pathological consequences to the host. This damage caused by the immune system is known as induced immunopathology. Immunopathology is caused by the excessive release of antibodies, interferons, and pro-inflammatory cytokines, activation of the complement system, or hyperreactivity of cytotoxic T cells. Secretion of interferon and other cytokine can trigger cell damage. Incubation period. Virus display variable incubation period upon virus entry into the host. The incubation period refers to the time taken for the onset of the disease after first contact with the virus.
Viral genetics. What is important in the survival of virus in nature? The capacity of replicate is the capacity to replicate rapidly, but if replication is too rapid, it can be self-defeating. The capacity to replicate rapidly, but if the replication is too rapid, it can be self-defeating. The capacity to replicate to high titer, like arbovirus, which give very high vertebrate host viremia. The capacity to replicate in certain tissue, the capacity to grow in immunologically sequestrated site, like brain, kidney, or in the cell of the immune system. The capacity to be shed for long period of time, intermittent shedding, is an additional advantage to the virus. And intermittent shedding is additional advantage to the virus. Define viral diarrhea, infection, and capillary. The capacity to elude host defense. Virus, especially those with large genome have genes that encode proteins capable of interfering with host and viral activity. Sheepdog virus immune tolerance is another mechanism in moving viral barrier. Cattle capacity to survive after being shed into the external environment. Canine parvovirus has environmentally stable capacity. The capacity to be transmitted vertically. Combined DNA technology, bacteria and plasmid isolated and gene inserted into the plasmid. After that, the plasmid is put in the bacterial cell. The cell multiply with gene for interest and copies of protein is produced. Protein used to make snow from a higher temperature. Protein dissolves blood clots in the heart attack already. Gene used to alter bacteria for cleaning up toxic waste. Gene for pest resistance inserted into plants. This is the application of this gambling DNA technology. Polymerase chain reaction. So this is the PCR, and this is its step. First, DNA is denatured, then DNA is denatured, continuing round of replication, another round of denature, primer attached, and the number of DNA that are doubled. Mutation. Depletion occurs in the coating of the viral nucleic acid that occur during replication are called mutation. However, a particular mutation is lethal to the virus. It depends on the result. Phenotypic characteristic virus-like technique will be used for study and isolation of the virus variant. This is a Blake assay. Types of mutation: genotypic mutation, also called a base substitution, occurs when a single nucleotide is replaced with a different nucleotide. A point mutation results in a base pair substitution after replication and mostly a mutant protein after transcription and translation. Parental DNA point mutation means difference in base pair. And this is the normal daughter DNA, and this is the mutant daughter DNA, and here the substitution of the base pair. A genotypic insertion mutation in which extra base pairs are inserted into a new place in the DNA. A deletion mutation in which a section of DNA is lost or deleted. Base substitution. Two main types of base substitution: transition during purine or pyrimidine to pyrimidine, transverse purine to pyrimidine or pyrimidine to purine. Transition are more. Insertion and deletion of deletion. Insertion or deletion of the one or two base change the purine frame. A frame or shift mutation error occurs at the DNA level, causing the codon to be parsed incorrectly. Next is mutation, in which substitution of a single base results in codon change, thus leaving a single AA substitution. Phenotypic changes. Condition lethal, multiply under some conditions but not others. Wild types grows under both sets of conditions. Temperature sensitive mutants don't grow at higher temperature. Host range mutants don't grow in all cell types that the phenotypic changes. A mutant may produce a different type of plaque in a cell. Layer, natural weak, viral fever virus mutant. Antibody scared mutant may become resistant to neutralization by anticoagulants against wild type in host persistently infected disease infectious anemia virus. A prominent example of mutation affecting viral antigenicity is the antigenic drift of influenza virus. Phenotypic changes of mutants grouped at elevated temperature than less susceptible to most fever response. Attenuated mutants, milder, no symptoms, vaccine development, pathogenesis, defective interfering virus demonstrated in most families, especially RNA virus, and in the presence of the wild type virus. Mutagenesis, spontaneous mutation, error in replication, transitions, and the transversions that occur during replication and can result from the amination of the cytosine. Here, the cytosine loss of amino group become uracil. Spontaneous lesions, debiorination, deamination, oxidative damage to DNA. Induced mutation, these analog mutagens, uracil, and substitute of T, and then one up binding with G result in miscoding. For example, uracil, ACT, and nucleotide are loads that can result in a chain termination. Is it induced mutation? Chemical agents such as nitrous oxide or nitroso-guanidine, ultraviolet ray, ionized radiation, cross-link between adjacent thymine dimer, site-directed mutagenesis, the nucleotide at any prescribed position in a DNA genome or complementary DNA not transcribed from an RNA genome can be changed to any other nucleotide or affected by the early insertion of an additional nucleotide. It can be done in bacteriophage, vector, vector, emitter. And here gene the vector, and then DNA polymerase, and then transform it, and here the E. coli host cells, and here the cell division. Genetic change according to some processes like mutation, recombination, exchange of information between two genomes, interaction between viral genes. This may involve not only the nucleic acid but also the proteins. The products of such interaction may affect the viral enzymes. Recombination, intramolecular recombination. It involves the exchange of nucleotide sequence between different but usually closely related viral gene replication in the coronavirus, and the poliovirus. Common DNA viruses. Recombination, genetic reassortment. Specifically RNA viruses, gene segmented genome, polyarity, fetal fever, and osmolarity, and disease. And the gluten influenza virus. Exchange of segments may occur when cells are infected, sometimes with two related viruses. Genetic change and recent evidence of viral evolution mechanism examining point mutation in influenza, intramolecular recombination between S9 and alpha virus, genetic reassortment, pandemic human virus, human influenza H1N1 in 1918 originated by genes from avian viruses, intramolecular recombination and mutation, polyvaccine virus, reversion to virulence following vaccination. Recombinant DNA technology. Box as a vector and recombinant DNA technology make vaccine. The large genome of virus has allowed the insertion of large amount of foreign DNA into non-essential genome regions. Okay. And there is veterinary vaccines prepared for vaccine. Recombinant vectors are generated by incorporating the foreign DNA into viral genome via homologous recombination. Proteins are expressed properly and undergone post-translation modification. Recombinant vaccine virus was used as rabies vaccine. Vaccine virus for rabies vaccine, and the Adragon is the invade rabies vaccine. The future of recombinant box virus technology, we can make multiple vaccines in one shot. That's the achievement of recombinant DNA technology. Complete sequencing of genome of the presented virus of all the families of DNA virus containing animal pathogens, characterization of the virus or viral DNA that are integrated into chromosomal DNA of transformed cells and animal infected with retroviruses, development of probes for use in rapid diagnostic assay to promoting the hybridization assay, application of PCR to amplify specific viral genome sequence for diagnostic and research purposes, production of protein coded by specific viral gene using bacterial yeast, bacteriovirus, and animal cell expression system. Epidemiology of viral diseases. Commonly used terms: incidence rate, attack rate, a measure of the occurrence of disease in a population over time, month or a year. It's used for describing acute disease of a short duration. Incidence rate, number of cases multiplied by 10, divided by the prevalence rate, variation at a particular point in time of number of cases currently present in the population divided by the number of animals in the population. Incidence rate, number of cases multiplied by 10 divided by population at risk at a particular point of time. The prevalence rate is thus a function of both the incidence rate and the duration of the disease, common terms morbidity rate. The percentage of animal or human in population that develop disease due to virus over a defined period of time. Mortality rate, the number of deaths from the disease in any given year divided by the total population at mid-year cause-specific mortality rate. Other terms and concepts used in epidemiology. In epidemiology, endemic disease, the continuous occurrence of disease in a population over a period of time. Endemic disease, the continuous occurrence of the disease in a population over a period of time. Epidemic disease refers to peaks in disease and symptoms. To pandemic disease refers to worldwide epidemics. Example: 1918 influenza pandemic. Epidemic disease refers to peak in disease incidence that exceed the endemic baseline in severity, potential economic impact. Virology because disease virus in poultry flock, or coronavirus in the human being in the recent days. Virus transmission cycle require entry into the body of a susceptible host, replication in its cells, shedding, spread to another host. Transmission cycle: entry, replication, shedding, spread to another host. Virus transmission Virus transmission may be horizontal between animals within the population at risk, or between humans within the population at risk. Vertical from dam to offspring, or from the mother to its daughter. And intertransmission. The transmission may occur direct transmission, licking, rubbing, beating, sexual contact. Indirect transmission, eating and drinking, containers, bedding, vehicles, syringe and needle, clothing, common vehicle, fecal contamination of feed and water supplies, airborne via droplet, foot-and-mouth disease. Horizontal transmission by arthropod transmission, iatrogenic transmission, multiple use of searing, non-sterile equipment, contaminated vaccine. Arthropod transmission by bites of mosquito, as Rift Valley fever, West Nile virus, Congo flu tongue virus. Nosocomial transmission occur in veterinary hospital. Disinfectant may be ineffective as in CDV. Zoonotic from animals to man. Rift Valley fever, West Nile virus. Arthropod vector, mosquitoes, Japanese encephalitis, dengue virus, yellow fever, Stewart-Lee's encephalitis, equine encephalitis, Western equine encephalitis, Venezuela, white encephalitis, Rift Valley fever, ticks, Congo hemorrhagic fever, various tick-borne encephalitis, sandy flies, Cilician sandy fly fever, Rift Valley fever, viral zoonosis, non-arthropod borne, herpes viridae, B virus, monkey, mode of transmission by animal bites. Here the virus family, here the virus, here the reservoir, and this is the mode of transmission. Herpes virus, B virus, monkey, the animal bite, mode of transmission. Both virus, virus, the virus, the reservoir, sheep and goat, mode of transmission, skin abrasion, rhabdoviridae, rabies virus, rats, skin abrasion, filoviridae, Ebola virus, monkey, bats, reservoir, rodents, iatrogenic nosocomial, morbillivirus, monkey, bats, rodents, iatrogenic nosocomial, orthovisoviridae, influenza virus, swine birth, respiratory, bonia viridae, hand-tan virus, rodents, contact with rodents and urine, anima virus, Lassa virus, rodents, and the mode of the animal bite here, the mode of transmission, contact with rodent urine. Arthropod virus genus, flavivirus, HV, rodents, the reservoir is the rodents, arthropod vector is the ticks, flavivirus, Rift Valley fever, mammals, the reservoirs, mammals, and arthropod vector mosquitoes, bonia viridae, the mammals and the ticks, bonia viridae, California encephalitis, mammals, the reservoirs, and the vector mosquitoes, Codivirus, the virus, Colorado tick fever, the reservoir is the mammals, and the vector is the tick. Arthropod virus transmission pattern, over 500 arbovirus are known, about 40 causes disease in domestic animals, some of which cause zoonotic diseases in man. Arthropod transmission may be mechanical, the virus restricted to the mouse part of the vector. Biological, the virus replicates in the tissue of the arthropod vector. In mosquitoes, the virus ingested with blood meal. Sufficient amount of the virus must be ingested, not less than 10 raised to 5 infectious units per ml. Virus multiplies in the gut epithelial cells. The virus leaks from the gut and infects the salivary glands, and the virus releases in the saliva during feeding. Cycle of Rift Valley fever infection. Here we find the mosquitoes, the vector, handling disease, amplification of care, and the valley fever cycle involving mosquito, animal, and the direct transmission to human. West Nile virus transmission cycle. West Nile bird as a reservoir host, and incidental infection occur in the animal, and incidental infections occur in human. Rift Valley fever emergency factor. Summary of Rift Valley fever emergency factor described and analyzed by Wilson. The economic development, the example or relative to Rift Valley fever, dams and irrigation, improvement, human demography and behavior, living with domestic animals, slaughter of sick animals, vaccination of healthy animals, international travel and commerce, domestic ungulate, export, human travel and migration, biological adaptation and change, increased viral virulence, improved vector competence, greater animal susceptibility, climate events, excessive rainfall. Vertical transmission, infections transferred from them to offspring before birth, avian reproductive virus, virus, bovine viral diarrhea, IDR. These infections may lead to immune tolerance, persistent by early embryonic death or abortion, congenital defect, congenital diseases. Here, bovine viral diarrhea as example of the vertical transmission. Here, subclinical, here clinical, mild, severe, death occur, and abortion of the calf, resistantly infected, immunotolerant, recurrent disease, occur in 40 to 120 days. Here, abortion occur in 90 to 160 days. Abortion or abnormalities, the antibody is positive or negative. More than 160 days, birth of abortion occur, or normal cause, antibody and immunity. Less than 40 days, embryonic disease.
Edward Jenner is considered the founder of vaccinology. In 1796, after he regulated a 13-year-old boy with vaccinia virus cowpox and demonstrated immunity to smallpox. In 1798, the first smallpox vaccine was developed. Origin of vaccines. Smallpox first disease people tried to prevent by purposely inoculating themselves with other infections. Inoculation started in India or China before 200 BC. Physicians immunized patients by picking off pieces from drying pustules of a person, inserting the powder into the person's nose. In 1718, Lady Mary Montagu reported that the Turks had a habit of deliberately inoculating themselves with fluid taken from mild cases of smallpox. Lady Montagu inoculated her own children in this manner. In 1796, Edward Jenner observed that milkmaids get infected with cowpox, a mild relatively of the deadly smallpox virus. Jenner took infectious fluid from hand of milkmaid Sarah Nelmes, inserted this fluid by scratching or injecting into the arm of a healthy local 8-year-old boy, James Phipp. Phipp then showed symptoms of cowpox infection and recovered after 48 days. Jenner injected some smallpox-infected matter into Phipp's, didn't later show signs of smallpox infection. Germs are all around us, both in our environment and in our bodies. Vaccination is a method of giving antigen to stimulate the immune response through active immunization. A vaccine is an immunobiological substance designed to produce specific protection against a given disease, viable or non-viable pathogens, or purified product. A vaccine is antigenic but not pathogenic. Types of vaccine: live vaccine, attenuated live vaccine, inactivated killed vaccine, surface antigen recombinant vaccine, toxoid, polysaccharide and peptide cellular fraction vaccine, live attenuated vaccine. Virulent pathogenic organisms are treated to become attenuated, retain their immunogenicity. Attenuated microbes reproduce in the recipient more robust and long-lasting immune response. Should not be administered to persons with suppressed immune response. Risk of a reverting to an active pathogen. Sabine poliovirus, measles, mumps, rubella, varicella, inactivated kill vaccine. Organisms are killed, inactivated by heat or chemical, remain antigenic, usually safe but less effective. No risk of vaccine-associated infection, more heat stable, only absolute contraindication, severe local or general reaction to previous dose. Microbial extract, stock of whole organism, it's composed of antigen globules, extracted from the pathogen or prepared by recombinant DNA technique. Efficacy of this vaccine varies. Toxoid, prepared by detoxifying the exotoxin of some bacteria, rendering them antigenic but not pathogenic. Adjuvant aluminum precipitation is used to increase the potency of the vaccine. When a person is susceptible and they encounter a harmful organism, it can lead to disease and death. The body has many ways of defending itself against pathogen, disease-causing organisms, skin, mucus, and cilia, microscopic hairs that move debris away from lungs, all work as a physical barrier to prevent the pathogen from entering the body in the first place. When a pathogen does infect the body, our body's defense called the immune system are triggered and the pathogen is attacked and destroyed or overcome. The body natural response to pathogen is a bacterium, virus, parasite, or fungus that can cause disease within the body. Each pathogen is made up of several subparts, usually unique to that specific pathogen and the disease it causes. The subpart of the pathogen that causes the formation of antibodies is called an antigen. The antibodies produced in response to the pathogen antigen are an important part of the immune system. You can consider antibodies as the soldiers in your body defense system. Each antibody or soldier in our system is trained to recognize one specific antigen. We have thousands of different antibodies in our bodies. When the human body is exposed to an antigen for the first time, it takes time for the immune system to respond and produce antibodies specific to that antigen. Once the antigen-specific antibodies are produced, they work with the rest of the immune system to destroy the pathogen and stop the disease. Antibodies to one pathogen generally don't protect against another pathogen, except when two pathogens are very similar to each other, like cousins. Once the body produces antibodies in its primary response to an antigen, it also creates antibody-producing memory cells, which remain alive when after the pathogen is defeated by the antibodies. If the body is exposed to the same pathogen more than once, the antibody response is much faster and more effective than the first time around because the memory cells are ready to pump out antibodies against that antigen. This means that if the person is exposed to the dangerous pathogen in the future, the immune system will be able to respond immediately, protecting against disease. This is existing pathogen and the existing antibody. It differs. This is the chicken pox. This is the measles, flu. Different pathogens have different shape, so different antibodies detected. How vaccine help? Vaccine contain weakened or inactive parts of the particular organism antigen that trigger an immune response within the body. Newer vaccine contain the blueprint for producing antigen rather than the antigen itself. Regardless of whether the vaccine is made up of antigen itself or the blueprint, or so that the body will produce the antigen, this weakened version will not cause the disease in the person receiving the vaccine, but it will prompt their immune system to respond much as it would have on its first reaction to the actual pathogen. The vaccine, and this is the new antibody. Vaccine is a teeny weakened, non-dangerous fragment of the organism and includes parts of the antigen. It's enough that our body can learn to build the specific antibody. Then in the body, you contour the real antigen later as part of the real organism. It already know how to defeat it. Some vaccines require multiple doses, even weeks or months apart. This is sometimes needed to allow for the production of long-lived antibodies and develop memory cells. In this way, the body is trained to fight the specific disease-causing organism, build up memory of the pathogen so as to rapidly fight it if and when exposed in the future. In the early century, 19th century, polio was a worldwide disease paralyzing hundreds of thousands of people every year. By 50s, two effective vaccines against the disease had been developed, but vaccination in some parts of the world was still not common enough to stop the spread of polio, particularly in Africa in the 1980s. A united world effort to eradicate polio from the planet began over many years and several decades. Polio vaccination using routine immunization visit and mass vaccination campaign has taken place in all countries. Millions of people, most children, have been vaccinated, and in August 2020, the African continent was certified wild poliovirus free, joining all other parts of the world except Pakistan and Afghanistan, where polio has not yet been eradicated. The success of an inactivated killed polio vaccine developed by Jonas Salk was announced in 1955. Another attenuated live oral polio vaccine was developed by Albert Sabin and came into commercial use in 1961. And this is a schedule, the national schedule for the vaccination of children from 2 months to 14-16 years. Every year, the influenza virus presents a new challenge. Reassortment takes place between genes from the older cold adapted master strain provide attenuated properties and gene from the newer wild type variant. In the laboratory, host cells are infected with the wild and cold strain, broken down, releasing RNA and enzyme into the host cell. Replication in host cells of influenza RNA can generate many combinations of wild type and cold adapted nucleic strands. One particular combination of wild and cold strain RNA will yield identifying protein of the wild influenza virus with the general biology of the cold adapted strain. The resultant flu vaccine stimulates the immune system by mimicking wild type flu virus, including their surface antigen, hemagglutinin, and neuraminidase. COVID-19 vaccine causes the immune system to create antibodies to fight COVID. Use a harmless version of spike-like structure on the surface of the COVID-19 virus called S protein. The main types: messenger RNA vaccine. This type of vaccine gives your cells instructions for how to make the S protein found on the surface of COVID-19 virus. After the vaccination, your muscle cell begin making the S protein base and splay them on the cell. Vector vaccine. In this type of vaccine, material from COVID-19 virus is placed in a modified version of the different viral vector. The viral vector gives your cells instruction to make copies of COVID-19 S protein. Protein subunit vaccine. Subunit vaccine include only the parts of the virus that best stimulate your immune system. This type of vaccine contains harmless S protein. Once your immune system recognizes S protein, it creates antibodies and defensive white blood cells. Microbial diagnosis. Some infectious diseases are distinctive enough to be identified clinically. Most pathogens, however, can cause a wide spectrum of clinical symptoms in humans. Conversely, a single clinical syndrome may result from infection with any one of many pathogens. For example, influenza virus infection causes a wide variety of respiratory syndrome that can't be distinguished clinically from those caused by streptococci, mycoplasma, or more than 100 other viruses. Most often, therefore, it's necessary to use microbiological laboratory methods to identify a specific etiological agent. Diagnostic medical microbiology is a discipline that identifies etiological agent of disease. The job of the clinical microbiological laboratory is to test specimen for patient for microorganisms that are or may be a cause of illness and to provide information when appropriate about the in vitro activity of antimicrobial drug against the microorganism identified. Steps for the laboratory diagnosis, microbial diagnosis, the patient, symptoms in the patient, localized or systematic, characteristic signs and symptoms. We notice a presumptive clinical diagnosis. We take sample, either sputum, blood, stools, tissue biopsy, urine, swab, anaerobic sample. We test for these samples, microscopic examination, culture, biochemical test, serological test, genetic. We reach a definitive diagnosis, antimicrobial susceptibility testing. We treat the patient and notice the improvement. Diagnosis of viral disease, direct diagnosis, isolation, experimental animal inoculation, tissue culture, detection by enzymatic activity. Number 2, enzymatic activity, electron microscope, immunoelectron microscope, detection of the nucleic acid, the viral diseases, experimentally transmission to the animals to produce virus which can't be propagated in vitro, to study the pathogenesis of the virus infections, to test vaccine safety, but they are increasingly being discarded for the following reasons: limitation, breeding and maintenance of the animals infected with pathogenic virus is expensive. Whole animals are complex systems in which it's sometimes difficult to discern events. Results obtained are not always reproducible due to host variation. Unnecessary or wasteful use of experimental animal is increasingly regarded as morally questionable. Serological diagnosis, tissue culture. By using tissue culture, either primary or cell line specific, CBE must be detected in the grown culture. Cytopathic effect. Usually the cytopathic effect followed by specific identification using ELISA, IHP, IFA and RIA. Nature of the antigen-antibody reaction. Nature of antigen-antibody reaction. Look at the key concept: non-covalent bond, hydrogen bonds, electrostatic bonds, van der Waals bond, hydrophobic bonds, multiple bond, reversible bond. The affinity. Difference of reaction between a single antigen determinant and single antibody combining site. The affinity, attractive and repulsive factor. Avidity, the overall strength of the binding between an antigen with many determinant and the multivalent antibodies. Specificity, the ability of an individual antibody combining site to react with only one antigen determinant. The ability of the population of antibody molecule to react with only one antigen. Cross-reactivity, the ability of an individual antibody combined site to react with more than one antigenic determinant. The ability of the population of the antibody molecule to react with more than one antigen. Shared epitopes, similar epitope. Factor affecting measurement of the antigen-antibody reaction: affinity, avidity, antigen-antibody ratio, and physical form of antibody. Test based on antigen-antibody reaction. All tests based on antigen-antibody reaction will have to depend on the lattice formation, or they will have to utilize ways to detect small immune complexes. All tests based on antigen-antibody reaction can be used to detect either antigen or antibody. Agglutination test, agglutination or hemagglutination, when the antigen is particulate. The reaction of an antibody with the antigen can be detected by agglutination, clumping of the antigen. When the antigen is an erythrocyte, the term hemagglutination is used. The term agglutinin is used to describe antibodies that agglutinate particulate antigen. When the antigen is an erythrocyte, the term hemagglutininin is often used. All antibodies can theoretically agglutinate particulate antigen, but IgM, due to its high valence, is particularly good agglutinin and one sometimes interfere that an antibody may be the ancient IgM class if it's good agglutinate antibody. Qualitative agglutination test. Agglutination test can be used in qualitative manner to assay for the presence of an antigen or an antibody. The antibody is mixed with the particulate antigen, and the positive test is indicated by the agglutinate of the particulate antigen. A patient red blood cells can be mixed with antibody to blood group antigen to determine a person's blood type. Agglutination reaction. Heme agglutination. The basic principles of heme agglutination assay is formation of clumps, agglutination of small particles coated with antigen when exposed to antibody specific for the antigen. Combined antigen antibody allow them to bind. The antigen is either a native constituent of the particle or is chemically attached to a particle. Antibody is added either on a solid or an agar or in a tube. Detect antigen antibody binding. The particulate will form a visible clump in antigen antibody binding where or the particle will remain dispersed if antigen antibody reaction occur. Agglutination reaction. Particles. There are three types of particles commonly used in agglutination reaction. Erythrocyte RBCs, bacterial cell often stained to make the clumps visible. Latex particles. With latex particles, the antigen are chemically attached prior to running the test. Applicant. Blood grouping. A view of the bed. This is blood typing. Anti-globulin Combs test. Direct anti-globulin Combs test. The reagent. Anti-globulin to canine antigen. Patient sample. Positive sample. Patient antigen present on RBCs. No patient Ig present. Here the agglutination. Positive anti-globulin causes RBCs to agglutinate, indicating presence of patient Ig on RBCs. Indirect anti-globulin Combs test. A normal RBCs patient sample serum, positive sample and negative sample, and this is a positive result, negative result. Combs test. In the direct anti-globulin test, the patient erythrocytes are tested for the presence of the patient antibody already bound to them. In an indirect anti-globulin test, the patient serum is tested for the presence of antibody that can bind to erythrocyte of normal. Latex agglutination test. Rheumatoid factor. Latex agglutination test. Rheumatoid factor. Two latex agglutination test. Positive test, clumping of the latex particle. The bottom, negative result. Covalent attached. The antigen IgG is covalently attached to the latex particle. In this test, presence of rheumatoid factor antibodies to IgG of the same animal is detected by clumping of the latex particle. Antibody to bacterial antigen. Hemagglutination inhibition test. Some viruses are capable of binding to an agglutinating erythrocyte of particular species of mammal, usually different from that of the natural host. The basic principle of the heme agglutination inhibition test is to inhibit inhibition of the heme agglutination induced by these viruses. Combined antigen antibody allow them to bind. The antigen and the antibody are mixed together in the tube or micro titer well. Detect antigen antibody binding. RBCs of appropriate species are added. If antibodies were present, RBCs are not agglutinated because antibody inhibit binding to the RBCs. If the antibody were absent, RBCs are agglutinated by the virus. Example: equine encephalomyelitis, influenza virus, Newcastle disease virus, paramyxovirus, parvovirus, canine porcine, and parainfluenza 3. Principle of HI. This is the positive and negative results. We test serum without antibodies, with or without antibodies. The RBCs of appropriate species, virus surface glycoproteins. This is the positive result, and this is the negative result. Heme agglutination inhibition test for Venezuelan equine encephalomyelitis virus. From left to right,
the result of the serum sample. Hem agglutination inhibition test for swine influenza from left to right, the result of the serum sample. Precipitation test, agar gel immunodiffusion, a plate is prepared with an agar gel into which antigen and antibodies can diffuse wells are punched into the agar gel, and antigen and antibody are placed into those wells. Sample being placed into a well on a gel that has been poured on the glass slide. As the antigen antibody diffuse toward one another in the gel, they set up concentration gradient and begin to combine. Agar gel immunodiffusion, a plate is prepared with an agar gel into which antigen and antibodies can diffuse. Wells are punched into the agar gel and the antigen and antibody are placed into these wells. Sample being placed into a well on a gel that has been poured on a glass slide. As the antigen and antibodies diffuse toward one another in the gel, they set up concentration gradient and begin to combine. In the region close to the antibody well, the small number of antigen present bind multiple antibody, the actual number of depending on the antigen size and the number of epitope, forming small complex that remain soluble in the region close to the antigen well. The small number of the antibody present bind to antigen, forming small in the region close to the antibody well. The small number of antigen present bind multiple antibody, the actual number depending on the antigen size and number of epitope, forming small complex that remain soluble. The region close to the antigen well, the small number of antibody present bind to antigens, forming small complex that remain soluble. Extensive cross-linking of antigen and antibody can occur, forming large complexes that precipitate in the gel. The precipitate that forms is visible as single white line, precipitate line in the gel, showed in dark gray in this single precipitate line in the gel. Multiple precipitate line if the antibody well has more than one antibody, which each antibody reacting with different antigen in the antibody well, then there will usually multiple precipitate line. Each antigen antibody reaction produce a separate precipitate line with the caveat that the line could overlap if the concentration were just right. The multiple lines form because each antigen antibody reaction is independent, that is, different molecular reactant combined to form different product. Line and antigen antibody identify a modification of the technique. Place multiple wells at angle to one another. If different sources of antibody are placed into wells with third well containing an antigen, it's possible to determine not only whether the antibody are present but whether they recognize the identical antigen. We make a modification by placing multiple wells at angles to differentiate between different antigens. Lines and antigen antibody defined. Lines of any identity top left. If the two antibodies do recognize the same antigen, a single precipitate line form, creating continuous between the two wells of antibody. Lines of non-identity. If the two antibodies recognize different antigen in the antigen well, then two precipitate lines form and cross one another. Lines of partial identity. Bottom
A line of identity forms for the reaction in common between two wells. A line of non-identity also forms next to the well with the extra antibody overlaps the line of identity. The resulting pattern appears as a line of identity with a spore is called a line of partial identity. Lines of identity on a slide. Lines of partial identity and non-identity, multiple antibody system, antigen antibody system combination between two pills, caprine arthritis, encephalitis virus, agar gel. A caprine arthritis, encephalitis virus antigen is placed in the center, and the outer will contain either control serum to contain antibody to CAEV. Clinical sample being tested. In this test, there is one positive sample indicated by the line of identity between it and the adjacent control wells. Agar gel for antibodies to fungal agents. This is commercially available kit for detection of presence of antibody to different fungal agent. The test on the left, coccidioides immatus. Enzyme-linked immunosorbent assay or ELISA. The basic principle of ELISA is to use an enzyme to detect the binding of antigen and antibody. The enzyme converts a colorless substrate chromogen to a colored product indicating the presence of antigen antibody binding. An ELISA can be used to detect either the presence of antibody or antigen in the sample depending on how the test is designed. Antibody detection. If the test is designed to detect antibody in a sample, a combine antigen and antibody allow them to bind. Antigen is firmly attached to the surface, usually a plastic well or filter. Sample being tested is added, allowed to incubate, then unbound antibodies are washed from the surface. Detect antigen antibody binding and antiglobulin C comment that is covalently attached to an enzyme. Enzyme linked is added, allowed to incubate, then unbound antiglobulin are washed from the surface. A colorless substrate of the enzyme is added. If the enzyme linked antiglobulin bound to the antibody on the surface, the enzyme will convert the colorless substrate to a colored product. ELISA to detect antibodies. The reagent, and this is a positive sample, the negative sample. Here is the reagent, enzyme labeled by antibody, specific for the antibody, allow time to react, wash away bound antibodies, and the reagent colorless substrate for the enzyme. Positive substrate converted to colored product. Antigen detection if the test is designed to detect antigen in the sample, combine antigen antibody, allow them to bind. An antibody that reacts with the antigen is firmly attached to the surface. Sample being tested is added, allowed to incubate, detect antigen antibody binding. Second antibody that react with the antigen and that is covalently attached to an enzyme linked is added, allowed to incubate, then unbound enzyme linked antibodies are washed from the surface. A colorless substrate of the enzyme is added. If the enzyme linked antibody bound to antigen on the surface, the enzyme will convert the colorless substrate to a colored product. Here is the steps. The agents, and this is the positive result. Substance converts to colored. Competitive ELISA. Competitive ELISA is a variation of ELISA that detect antibody in the sample. It's based on the principle that antibody in the sample will bind to an antigen and then inhibit binding of an enzyme linked antibodies that react with the antigen, compete for binding. Combine antigen and antibody, allow them to bind. The antigen is firmly attached to the surface, usually plastic well or filter. The Sample being tested is added and allowed to incubate, detect antigen antibody binding, and enzyme linked antibodies that react with the antigen is added, allowed to incubate, then pushed from the surface. Colorless substrate of the enzyme is added. A colorless substrate of the enzyme is added. If the sample contain antibodies that react with the antigen, they would inhibit binding of the enzyme labeled antibody, and no color reaction would occur. If the sample doesn't contain antibodies that react with the antigen, the enzyme labeled antibody would bind and the color reaction would occur. Note that in the competitive ELISA, no color reaction is a positive result and a color reaction is a negative result. And this is the competitive ELISA reagent and the positive negative. Radioimmunoassay. The basic principle of radioimmunoassay is the use of radio-labeled antibody or antigen to detect antigen-antibody reaction. The antibody or antigen are labeled with 125 iodine 125 isotope, and the presence of antigen-antibody reaction is detected using a gamma counter. Combine antigen-antibody allow them to bind. An antibody to the hormone is attached to a filter. The serum sample is added and any hormone antigen is allowed time to bind to the antibody. Detect antigen-antibody binding. A radio-labeled hormone is added and allowed time to bind. All unbound substances are washed away. The amount of bound radioactivity is measured in gamma counter. The presence of the hormone in the serum sample inhibits binding of the radio-labeled hormone. Thus, the amount of radioactivity present in the test is inversely proportional to the amount of hormone in the serum sample. A standard curve using increasing amount of known concentration of the hormone is used to determine the quantity in the sample. And this is reagent, antibody specific for hormone, coating the filter, unknown sample with the hormone, allow time to react, wash away unbound substance. The reagent, iodine 125 labeled hormone, allow time to react, wash away unbound radioactive hormone. Measure the radioactivity in a gamma counter. The result amount of radioactivity is inversely proportional to the concentration of the hormone in the sample. RIA are highly sensitive and quantitative, capable of detecting a small amount of antigen or antibody. As a result, they are often used to measure the quantity of hormone or drug present in the patient's serum. Often RIS are performed in solution rather than on filter. Antigen-antibody complex precipitate from the solution and the amount of radioactivity in the supernatant is measured. In this case, the amount of radioactivity present in the supernatant present the unbound radio-labeled hormone is proportional to the directly proportional to the amount of hormone in the sample. Immunofluorescence assay or fluorescent antibody test. The basic principle of the immunofluorescent assay is to use fluorescent compound, usually fluorescent, to detect the binding of antigen-antibody. The antibody is labeled with the fluorescent compound and its presence is detected using fluorescent microscope. Under fluorescence microscope, fluorescent appears bright green wherever the binding occurs. Another term for this assay is fluorescent antibody test or FAT. There are two types of IFA assay, direct and indirect. Direct FAT detect the presence of an antigen in the tissue. Indirect FAT detect the presence of antibodies in the serum or other fluid. Direct and indirect FAT. Direct FAT prepare the antigen. The antigen is a tissue section, cytology smear or impression smear. Combine antigen and antibody, allow them to bind. Add fluorescent label antibody that react with the antigen you want to detect, then a rise of the unbound antibody. Detect antigen-antibody binding. Observe the slide under a fluorescent microscope. Indirect FAT prepare the antigen. The antigen is tissue section from a healthy animal or a culture cell line. Combine antigen or antibody, allow them to bind. The patient's serum is added to the tissue or cells, then unbound antibodies are rinsed off. Detect antigen-antibody binding. Fluorescent labeled antiglobulin is added to the slide, then rinsed off. Observe the slide under a fluorescent microscope. And this is the reagent. Unknown sample fixed to the slide. Fluorescent labeled antibody to antigen histochemistry. The basic principle of immunohistochemistry is the use of enzyme-linked antibodies to detect tissue antigen. The colorless substrate is converted by the enzyme into colored product that precipitate on the slide at the site of the reaction. Thus, immunohistochemistry localizes antigen in a tissue section. Prepare the antigen, conventional histologic section, or fresh cytological smear or fresh impression smear from tissue. Combine antigen antibody, allow them to bind. Add the enzyme linked antibody to the slide. Incubate, then wash to remove unbound antibody. Detect antigen antibody binding. Add the colorless substrate. Incubate, then wash to remove remaining substrate. Counter stain to make non-staining cell and structure visible. If antigen were present in the tissue, a color reaction indicates its location in the cell or in the extracellular matrix. The tissue section is counter-stained to permit visualization of unstained cells. However, the architecture of the tissue section is not as well preserved as one would observe with a standard hematoxylin and iosine immunohistochemistry. This is the reaction, the reagent, and this is a positive and negative sample. The reagent enzyme labeled antibody to antibody of interest, allow time to react, wash away unbound antibody, colorless substrate for enzyme as reagent, positive substrate converted to colored precipitate at the site of the
Welcome. Sometimes diarrhea causes deaths in neonates, make these problems in different places in the world. An overview of the diarrheal viruses. Viral gastroenteritis. It's thought that viruses are responsible for up to 3/4 of all infected diarrhea. Viral gastroenteritis is the second most common viral illness after upper respiratory tract infection. In developing countries, viral gastroenteritis is a major killer of infants who are undernourished. Rotavirus are responsible for 500,000 deaths a year. Many different types of viruses are found in the gut, but only some are associated with gastroenteritis. Virus found in the gut associated with gastroenteritis: rotavirus, adenovirus, calcivirus, novac virus, small round structure virus, astrovirus, coronavirus, virus. Virus found in the gut and not normally associated with gastroenteritis: polio, coxsackie A, coxsackie B, echo, enterovirus, 68, 61, hepatitis E, adenovirus, reovirus, found in the gut as opportunistic infection, CMV, HSV, VZV, HIV, rotavirus particle. This is the shape of the rotavirus under the electron microscope. Rotavirus, naked double-stranded RNA virus, 80 nanometer in diameter, also found in other mammals and birds, causing diarrhea, account for 50 to 80% of all cases of viral gastroenteritis, usually endemic but responsible for occasional outbreaks. Causes diseases in all age groups, but most severe symptoms in neonates and young children. Asymptomatic infection common in adults and other older children. Symptomatic infection gain common in people over 60. Up to 30% of mortality rate is malnourished children, responsible for up to 500,000 deaths per year. Rotavirus, 80% of the population have antibodies against rotavirus by the age of 3. More frequently during the winter, fecal oral spread respiratory droplets. 24 to 48-hour incubation period followed by an abrupt onset of the vomiting and diarrhea. A low-grade fever is present. Diagnosis by electron microscope provide the detection of the rota antigen in feces, ELISA, or other assay. Live attenuated vaccine is not available for use. Adenovirus particle. This is the adenovirus particle in the electron microscope. Enteric adenovirus, naked DNA virus, 75 nanometer in diameter, fastidious enteric adenovirus type 4, and 41 are associated with the gastroenteritis associated with cases of endemic gastroenteritis, usually in young children, and can cause occasional outbreak. Possibly the second most common viral cause of gastroenteritis, 7 to 50%, 15% of all cases. Similar disease to rotavirus. Most people have antibodies against the enteric adenovirus by the age of 3, diagnosed by electron microscope or by the detection of adeno antigen in feces by ELISA or other assays. Astrovirus particle, and this is the astrovirus particle under the microscope. Astrovirus small RNA viruses named because of star-shaped surface morphology, 28 nanometer in diameter, associated with cases of endemic gastroenteritis, usually in the children and innate, can cause occasional outbreak, responsible for up to 10% of cases of gastroenteritis. Similar diseases to rota and adenovirus. Most people have antibodies by the age of 3, diagnosed by electron microscope only, often very difficult because of the small size. Calcivirus particle, and this is the particle shape under the electron microscope. Calcivirus is small RNA virus, characteristic surface morphology consisting of hollow particle 35 nanometer in diameter, associated mainly with epidemic outbreak of gastroenteritis, also occasionally responsible for the endemic cases like norovirus virus. Vomiting is a prominent feature of the disease. Majority of the children have antibodies against calcivirus by the age of 3, diagnosed by electron microscope only, often difficult to diagnose because of the small size. Norovirus-like virus, Norovirus-like virus particle. Norovirus-like viruses, small RNA virus with ragged surface, 35 nanometer, now classified as calcivirus, always associated with epidemic outbreak of gastroenteritis, other more commonly affected children, associated with consumption of shellfish and other contaminated food, results spread possible as well as fecal oral scrape. Also known winter vomiting disease, with vomiting being the prominent symptoms. Diarrhea usually mild, antibody acquired later in life. In US, only 30% of the adults are seropositive by the age of 50. Diagnosis is made by electron microscopy and by PCR. Other possible diarrheal virus, coronavirus, RNA virus with a crown-like appearance, conveniently associated with gastroenteritis at present. Small virus like particle with a small surface, 22 to 28 nanometer in length, may possibly be the parvovirus, enterovirus, cubic bacteriophage, occasionally seen in the fecal pieces of endemic or epidemic cases of gastroenteritis. Respiratory viruses associated with respiratory infection syndrome, commonly associated with virus, influenza, rhinovirus, coronavirus, less commonly associated with virus, influenza, influenza syndrome, influenza like symptoms, influenza virus, less commonly with paramyxin virus, virus, respiratory virus, bronchitis, RSV, respiratory CCP virus, associated with influenza, bronchopneumonia, influenza virus, respiratory CCP virus, influenza virus, RNA virus, genome, processes with ACN, enterovirus with urine disease spikes. Three types, A, B, and C. Type A undergo antigen shift detect, type B undergo antigen shift only. Type C is relatively stable. Influenza A virus undergoes antigen shift and antigen drifts. These three pathogens and monomer proteins antigen shift to the active pathogen result in antigen drift. In the H and N protein will result in epidemics, usually caused mild febrile illness. Usually caused mild febrile illness. Death may result from complications such as viral bacterial pneumonia. Epidemiology, pandemics, influenza E pandemics arise when a virus with a new antigen subtype emerge as a result of antigenic shift. As a result, the population has no immunity against the new strain. Antigenic shift had occurred 3 times in the 19th century. Pandemics for the A and B arise in more neo-antigenic drift as a result of mutation. Vast antigenic shift in 1918. H1N1 Spanish influenza leading to 14 million deaths. In 1957, H2N2 Asian flu 1 to 2 million deaths. In 1968, H3N3 Hong flu 700,000 deaths. In 1977, H1N1 big emergence in Europe pandemic. At least 15 or 16 HA subtypes and 9 NA subtypes appeared in nature. 1997, where 18 also were infected with which 6 died. The source of the virus was reported from infecting chicken, and the outbreak was initially controlled by gnats smothered in the chicken in the turkey. All strains of the infected virus were totally alien in origin, and there was no disease of the assortment. However, the strains involved were highly virulent for their natural agent. H9N2, several cases were infected with the H9N2 virus appeared in Hong Kong and the China in 1999. The disease was too mild. All patients made a complete recovery against the usual disease of the assortment theory. The shift the assortment of the H and N genes between human and the alien influenza virus to a third group. There is good evidence that this occurred in 1957 H2N2 and in 1968 H3N2 pandemic. Recycling of pre-existing strains is probably occurred in 1977 when H1N1 surfaced. Gradual adaptation of alien influenza virus to human transmission. There is some evidence that this occurred in 1918 H1N1 pandemic. So avian H3 and H2, the assortment in the leak and reinfection of human as H3, laboratory diagnosis, detection of pharyngeal, rapid diagnosis can be made from the detection of human pharyngeal from the pharyngeal aspirate through motion by IFT or ELISA. By circulation, virus may be readily outbreak with nasopharyngeal aspirate and include swab. Serology, a retrospective diagnosis is claimed by serology tests sympathy, most widely used, HER and ELISA may be used to give a type-specific diagnosis. The management amantadine, heparin, neuraminidase inhibitors used prevention inactivated split subunit vaccines are available against influenza. They use the vaccine is normally trivalent, consisting of 1A H3N2 strain, 1A H1N2 strain, and 1B strain. The strains used are reviewed by WHO. The vaccine should be given to individuals and left by individuals who are at risk of severe infection. Amantadine can be used as an prophylaxis for those who are allergic to the vaccine or to the before the vaccine takes effect. Parameter virus. Single-stranded RNA virus includes the polymorphic methylation 5 subtypes 1, 2, 3, 4, 5, and 4, 4A and 4B. No common convention, also due to Mumps virus. Manifestation, group laryngeo tracheobronchitis, most common method of coronavirus infection. However, other virus may group and influenza in this virus is a virus. Other conditions that may be caused by coronavirus include colitis, pneumonia, like tracheobronchitis, and peritonitis illness. Lab diagnosis, infection of the antigen. The rapid diagnosis can be made by detection of the coronavirus influenza antigen in the nasopharyngeal aspirate precaution. Virus isolation may be used check by swab, serology, retrospective diagnosis is made by serology CRP, most widely management, antiviral, comparable available. These cases of group should be admitted to hospital, placed with hospital tents, no vaccine is available. Characterizing syncytial virus, RSV, single-stranded RNA in virus. belongs to the genus virus of the virus family. Considerable strain distribution of this will be classified into subgroups A, B5, both subgroups circulate in the community as line causes and sizable each year. Encapsulation in terms of severe lower respiratory tract disease and new factors for 50 to 90 bronchiolitis, 5 to 40 bronchopneumonia. Other implications include growth or 10% of all cases. In older children and others, the symptoms are much milder. It may cause epicardial illness or bronchitis. Infants at risk of severe infection, infants with congenital heart disease, infants who were hospitalized in the first days of life with congenital disease are particularly at risk. Infants with underlying pulmonary disease, infants with underlying pulmonary disease, especially bronchoalveolar dysplasia, are at risk of developing prolonged infection with this particular syncytial virus. Immunocompromised infants, children who are immunocompromised or have congenital immunodeficiency disease may develop lower respiratory tract disease and each infection. Antigen rapid diagnosis can be made by detection of respiratory disease like virus antigen from nasopharyngeal aspirate. Rapid diagnosis is important because of availability of therapy. Virus inflation. Virus may be readily isolated from the nasopharyngeal aspirate, however this may take several days. Serology, a retrospective diagnosis may be made by serological CRT most widely used. Aerosolized repatriation used for infant infection and for those at risk of disease. There is no available respiratory syncytial virus infection can be used to detect infant at risk of respiratory syncytial virus. Adenovirus, double-stranded DNA virus, non-lethal at least 47 non-classified disease, sufficient immunity, hereditary assembly, pharyngitis conjugated, pharyngeal conjunctival fever, immunity, respiratory disease of 3 weeks, over 14.1, pneumonia, follicular conjunctivitis, epidemic, keratoconjunctivitis, 37, diffuse like cerebral bile acute hemorrhagic cystitis, 1 acute infantile gastritis, 14 to 41. Interceptions 1 to 5, severe disease in the other immunocompromised patient 5, 34 to 35, meningitis 3 to 7, regular diagnosis, detection of the antigen, and diagnosis can be made for detection of adenovirus antigen from nasopharyngeal aspirate and throat washing, virus isolation, virus isolation, nasopharyngeal aspirate, throat swab, and serology, and retrospective diagnosis may be made by serology, the most widely treatment. There is no specific antiviral therapy. Vaccine is available and used other respiratory syndrome that consists of live adeno 4 taken with capsules, the weeks in various hours, 14 around week. All virus will account for 1/3 to 1/2 of all respiratory infections. Real virus are responsible for 30 to 50% of the common cold coronavirus, 10 to 30%. The rest are the virus, including virus, virus, influenza, and parental which may cause symptoms distinguishable from those of the virus. Single-stranded RNA virus belongs to the recorded virus family. Two-stranded RNA virus acting about at least 165 abnormal coronavirus. Single-stranded RNA virus involves polymorphic morphology, serotypes, and there is new emerging virus, SARS, and COVID-19. Welcome. Herpes virus. Properties of the herpes virus: envelope, double-stranded DNA virus, genome consisting of long and short fragments which may be oriented in either direction, giving a total of 4 isomers. 3 subfamilies: alpha herpes virus, HSV1, HSV2, VZV, herpes simplex, beta herpesvirus, CMV, HHV6, HHV7, gamma herpesvirus, EBV, HHV8. Set up latent or persistent infection following primary infection. Reactivation are more likely to take place during the period of immunosuppression. Both primary infection and reactivation are likely to be more serious in immunocompromised patients. Herpesvirus particles. HSV2 virus particle. Note that all herpesvirus have identical morphology and can be distinguished from each other under electron microscope. Herpes simplex virus belong to the alpha herpesvirus subfamily of herpesvirus, double-stranded DNA enveloped virus with a genome of around 150 kilobytes. The genome of HSV and HSV2 share 50 to 70% homology. They also share several cross-reactive epitopes with each other. There is also antigenic cross-reaction with VZV. Man is the only natural host for HSV. Virology. HSV is spread by contact, and the virus is shed in saliva, tears, genital, and other secretions. By far the most common form of infection results from a kiss given to a child or adults from a person shedding the virus. Primary infection is usually trivial or subclinical in most individuals. It's a disease mainly of very young children, those below 5 years. There are two peaks of incidence: the first 0 to 5 years, and the second in the late teens when sexual activity commences. About 10% of the population require HSV infection through the genital route, and the risk is concentrated in young adults. Virology. Generally, HSV1 causes infection above the throat and HSV2 below the belt. In fact, 40% of the clinical isolate from the genital source are HSV1, and 5% of the strains isolated from the facial area are HSV2. This data is complicated by oral sexual practices. From primary infection, 45% of the oral infected individuals and 60% of the patients with genital herpes will experience recurrence. The actual frequency of recurrence varies widely between individuals, and the mean number of the episodes varying about 1.6. Pathogenesis. During the primary infection, HSV is spread locally and a short-lived viremia occurs, whereas the virus is disseminated in the body, spread to the craniospinal genital vein, established latency in the craniospinal ganglia. The exact mechanism of latency is not known. It may be true latency where there is no viral replication or viral resistance, where there is a low level of viral replication. Reactivation. It's well known that many triggers can provoke a recurrency. They include physical and psychological stress, infection, especially pneumococcal and meningococcal, fever, radiation, including sunlight, and masturbation. Clinical manifestation. HSV will involve in a variety of clinical manifestation, which is acute gingivitis, herpes labialis, cold sore, ocular herpes, herpes genitalis, other forms of cutaneous herpes, meningitis, encephalitis, neonatal herpes, oral facial herpes, acute gingivitis. Acute gingivitis is the commonest manifestation of the primary hepatic infection. The patient experiences pain and bleeding of the gum, 1 to 8 mm ulcer with necrotic bases or present. Neck glands are commonly enlarged, accompanied by fever, usually a self-limiting disease which lasts around 13 days. Herpes labialis, cold sore, following primary infection. 45% of the oral infected individual will experience reactivation. The actual frequency of recurrence varies widely between individuals. Herpes labialis, cold sore is a recurrence of oral HSV, a prodrome of tingling, coldness, and itching at the site usually heralds the recurrency. About 12 hours later, fevers disappear following the bipolar papules, and then vesicles. Gingivostomatitis. Ocular herpes. HSV causes a broad spectrum of ocular disease, ranging from mild to superficial lesion involving the external eye to severe sight-threatening disease for the inner eye. Diseases caused include the following: primary HSV keratitis, dendritic ulcers, recurrent HSV keratitis, HSV conjunctivitis, iridocyclitis, carrying genital herpes. Genital lesion may be primary, recurrent, or initial. Many sites can be involved, which include the venous, vagina, cervix, anus, vulva, bladder, and the sacral nerve roots, the spinal, and the meninges. The lesions of genital herpes are particularly prone to the secondary bacterial infection, like Staphylococcus, Streptococcus, Trichomonas, and Candida albicans. Dysuria is a common complaint in severe cases, may be urinary retention. Local sensory nerves will be involved, leading to the development of radiculitis, a mild meningitis may be present. 60% of the patients with genital herpes will experience recurrent lesions in the perianal area tend to be more numerous and persist longer than the oral HSV1 counterparts. Herpes simplex encephalitis. Herpes simplex encephalitis is one of the most serious complications of the herpes simplex disease. There are two forms: neonatal, there is global involvement, and the brain is the most liquefied. The mortality rate approach 70%. Focal disease, the temporal lobe is most commonly affected. This form of disease appears in children and adults. It's possible that many of these cases arise from reactivation of virus. The mortality rate is high, 70% without treatment. It's of utmost importance to make a diagnosis of herpes simplex early. It's generally practiced that IV acyclovir is given in all cases of suspected HSE before laboratory results are available. Neonatal herpes simplex. Incidence of neonatal herpes simplex infection varies from country to country. The baby is usually infected primarily during passage through the birth canal. Premature rupture of the membrane is a clearly recognized risk factor. The risk of neonatal transmission is greatest when there is a florid primary infection in the mother. There is an appreciably smaller risk from recurrent lesion in the mother, probably because of the lower viral load and the presence of specific antigen. The baby may also be infected from other sources such as oral lesions from the mother or herpetic white blood in the uterus. The neonatal herpes simplex is the spectrum of neonatal infection varies from mild disease localized to the skin to a fetal disseminated infection. Infections is particularly dangerous in premature infants, where dissemination occurs. The organ most commonly involved are the liver, adrenals, and the brain. Where the brain is involved, the prognosis is particularly severe. The encephalitis is global and with such severity that the brain may be liquefied. A large proportion of the survivors of neonatal HSV infection have residual disabilities. Acyclovir should be properly given in case of neonatal HSV infection. The only means of prevention is to offer cesarean section to mother with large genital HSV. Dissemination herpes simplex are much more likely to occur in immunocompromised individuals. The widespread vesicular resembles that of chicken pox. Many organs other than the skin may be involved in liver, spleen, lung, and CSF. Other cutaneous manifestations include eczema, hypericum, which is facial eczema disease that occurs in a patient with eczema. Herpetic white flow, which arise from implementation of the virus into the skin and typically affect the fingers. Zoster 4 herpes simplex. This is a rare presentation of herpes simplex where HSV lesions appear in the dermatomal distribution similar to herpes zoster. Lab diagnosis: electromicrograph, electron microscope, vesicle fluid, fragment result, but can distinguish between herpes zoster and herpes simplex. Immunofluorescence skin scraping can distinguish between both of them. PCR used routinely for diagnosis of herpes simplex. Virus isolation: HSV1, HSV2 are among the easiest viruses to calculate. It usually takes 1 to 5 days for results to be available. Serology: not that useful in the acute phase because it takes 1 to 2 weeks before and it will appear after infections. Used to document recent infection. This is herpes simplex infected epithelial cell in the skin, and this is the cytoplasmic effect of HSV in cell culture. Management: There are only a few indications of antiviral chemotherapy, with the high cost of antiviral drug being a main consideration. Generally, antiviral chemotherapy is indicated where the primary infection is especially severe, where there is disseminated, where site is threatened and herpes simplex encephalitis. Where there is dissemination, site is threatened and herpes simplex encephalitis. Acyclovir, we use acyclovir and famine. Acyclovir, valprocyclovir,